Good evening, students. Back to botany classes. In yesterday's class, I explained you about the pollen viability. After that, I explained you about the pistil. What are the parts of pistil? I explained about the syncarpus and apocarpus. After that, I explained you about megasporangium or ovule. So, what are the different parts of megasporangium? So, there I explained you anatropous ovule, vertical section of an anatropous ovule. Uh, Anatropous ovule, it is a inverted ovule. Their micropyle and funicle micropyle and chalaza lie in same line, but funicle do not. Such type of ovule is called as anatropous ovule. What are the parts of a typical ovule? First one, funicle. Stalk of the ovule is called funicle, by which it is attached to the placenta. Second one, hilum. The point of attachment of funiculus to the ovule is called as hilum. And one more thing, in anatropous ovule, funicle continue, it will grow to form a ridge called as raphe. Next one is integument. Ovule is covered by one or two protective pores called integument. Some the ovule is covered by a single integument called as unitegmic. In most of the member, ovule is covered by two protective pores called as bitegmic. Rarely in some, ovule is covered by three protective pores called as tritegmic. It is even. It is this third integument is called as edile in the member of Asphodilus. Is it clear? Integument do not completely cover, it leave a small opening, and that opening is called as micropyle. Through this micropyle, the pollen tube enter into the embryo sac. Next one is the region where integument starts or begins. That region is called as the chalazal region or chalaza. And moving to the next one is the nucellus. It is a massive parenchymatous tissue. It stores reserve food material. Nucellus cells are diploid. They are not haploid. Cells are parenchymatous and diploid. Coming to the last one, embryo sac. In angiosperm, female gametophyte is commonly called as embryo sac. It is formed by the one of the nucellus cell functions as megaspore mother cell. When that megaspore mother cell undergo meiotic division, results in the formation of four megaspores. Commonly, out of four megaspores, only one megaspore take part in the development of embryo sac. So commonly, each ovule contains single female gametophyte or single embryo sac. And embryo sac, it is a 7-celled and 8 nucleate. So in yesterday's class, I explained the anatropous ovule in detail. So beside that, other types of ovules are also there. Orthotropous ovule, hemianatropous ovule, compylotropous ovule, and amphitropous ovule and synopsis ovule. So these are the different types of ovules are there. So in yesterday's class, still I explained you about the compylotropous ovule. So after that, we'll go to the next one is the amphitropous ovule. So in amphitropous ovule, in this type of the ovule, both embryo sac and the ovule is ovule horseshoe shape, and this type of the ovule is called as the amphitropous. So in this member, so the embryo sac as well as the ovule curved as a horseshoe. Curved as a horseshoe. Such type of the ovule is called as the amphitropous ovule. Example, you have to take down the copy. So this is an example for the amphitropous ovule. Moving to the last type of the ovule, that is the synopsis ovule. Synopsis ovule. Cystinotrophus ovule. So in this type of ovule, the funicle, it curved to 360-degree angle, curved 360-degree angle. Here funicle completely coiled the ovule. This is a funicle. So this funicle, it is completely coiled the ovule. Such type of the ovule is called as the Cystinotrophus ovule. Is it clear? And this we are going to observe in Opuntia. So these are the different types of ovules. So how many types of ovules are there? There are 6 different types of ovules are there. One is the orthotropous, second one anatropous, third one hemianatropous, fourth one comphinotropous, fifth one amphitropous, and the last one is the cystinotrophus. So this is the brief introduction about the types of ovules. So after this we will go to the next one. Moving to the next, Megasporogenesis. Moving to the next one is the Megasporogenesis. So what is meant by the Megasporogenesis? So before that, I told you that inside the integument there is a massive parenchymatous tissue called as nucellus. So many cells are present, but one of the nucellus cell present near micropylar region. One of the nucellus cell present near micropylar region functions as megaspore mother cell. This megaspore mother cell or this cell, it different from other cells. Cell is large, it contains dense cytoplasm and prominent nucleus. Is it clear? One of the nucellus cell functions as megaspore mother cell. The cell which is present near micropylar region or chalagial region. One of the nucellus cell present near micropylar region functions as megaspore mother cell. Actually, nucellus region, so many parenchyma cells are there. Is it clear? Here we are going to observe in detail nucellus are of 2 types. One is the crossy-nucellate and second one is the teny-nucellate. Crossy-nucellate and second one is the teny-nucellate. Crossy-nucellate means nucellus well developed. Nucellus is well developed. That is called as the crossy-nucellate. Teny-nucellus means nucellus poorly developed. So nucellus are 2 types on the basis of the development. One is crossy-nucellate and second one teny-nucellate. So crossy-nucellate, here nucellus is well developed, that is called as a crossy-nucellate. Teny-nucellate means nucellus is poorly developed. Here, in crossy-nucellus, what will be happen? Nucellus means hypodermal region. That is present near the micropyle. So, micropylar region, cell act as a archisporion. archisporial. Cell which is present near the micropylar region functions as the archisporial. This archisporial divides to form two cells. It divides two cells. Is it clear? One is the primary parietal. It divides periclinally to form two cells. One is the primary parietal cell and the second one is the sporogenous. Primary parietal and second one is the sporogenous. And this sporogenous, what it develops? It develops the megaspore mother cell in the member of crossinucleate. Crossinucleate. Is it clear? In crossinucleate, what will be happen there? So the hypodermal of nucellus cell, which is present near the micropylar, functions as the archisporial. So that divide periclinally to form two cell, primary parietal as well as the sporogenous cell. That sporogenous cell functions as the megaspore mother cell. And in perinucleate, what will be happen there? Directly, one of the nucellus cell that directly functions as megaspore mother cell. So that megaspore mother cell, large dense cytoplasm and a prominent nucleus. So here, what is meant by the megasporogenesis? Megasporogenesis means what is meant by the megasporogenesis? The process of formation of megaspores from megaspore mother cell through meiosis is called megasporogenesis. So what is meant by the megasporogenesis? Megasporogenesis means the process of formation of megaspores from megaspore mother cell through meiosis is called megasporogenesis. So megaspore mother cell, it is a 2N. What the ploidy of megaspore mother cell? So the ploidy of megaspore mother cell is 2N. When this megaspore mother cell, when it undergoes meiotic division, results in the formation of tetrad of megaspores. It means that 4 megaspores are formed. Is it clear? So what is the ploidy of the MMC? It is a 2N. Then what is the ploidy of nucellus? Ploidy of nucellus. What is the ploidy of nucellus? The ploidy of nucellus is also diploid. The ploidy of nucellus is also diploid. Remember it here. What is the ploidy of megaspore mother cell? The ploidy of megaspore mother cell is diploid. It is not a haploid, it is a diploid. So this diploid cell undergoes meiotic division to form what? tetrad of megaspore. So what is the ploidy of nucellus? So the ploidy of nucellus is the diploid. Is it clear? So here this is the megaspore mother cell. Megaspore mother cell. So this megaspore mother cell undergoes which division? Meiotic. So in first meiosis, what will happen there? How many cells are formed? 2 cells. In the first meiotic division. So what will happen there? 2 megaspore, this is called as dyad. Dyad means 2 megaspore. So in meiosis 2, how many megaspores are formed here? 4 megaspores. So these are the tetrad of megaspores. Tetrad of megaspores. Is it clear? So you know very well that at the end of mitosis, how many daughter cells are formed? 2 daughter cells are formed. But at the end of the meiosis, how many daughter cells are formed? 4 daughter cells are formed. So when megaspore mother cell, when it undergoes meiotic division, it results in the formation of what? Tetrad of megaspore. So this is the nucellus. Nucellus. And this is the one of the cell of the nucellus which functions as megaspore mother cell. So it undergoes meiosis to form 2 daughter cells in first meiosis 1, and in second meiosis there is a formation of tetrad of megaspores. And here a very important one is here, these megaspores are generally arranged in linear. They are arranged in linear. While explaining microsporogenesis, I told that the microspore tetrad generally they are arranged in tetrahedrally or isobilaterally. But in megasporogenesis, megaspores are arranged generally linear fashion. Is it clear? So this stage is called as the megasporogenesis. Megasporogenesis. Okay, now we are going to study about the megametophyte, or we can tell that is known as the female gametophyte. Okay. After this, we'll go to the female gametophyte. Moving to the next one is female gametophyte. Female gametophyte. Till now you studied that formation of the megaspores, how megaspores are formed. Megaspores are formed when megaspore mother cell undergoes meiotic division. Is it clear? So now the female gametophyte. So how the female gametophyte or embryo sac is formed? Is it clear? Here, scientist P. Maheshwari classified 3 different types of embryo sac. Classified 3 types of embryo sac on the basis of number of megaspores. Scientist P. Maheshwari classified embryo sac into 3 types on the basis of number of megaspores take part in the development of embryo sac. What are those 3 different types of the embryo sac? One is monosporic. Second one is bisporic, and the third one is tetrasporic. So embryo sac classified into 3 types. One is monosporic embryo sac, second one bisporic embryo sac, and the third one is the tetrasporic embryo sac. Monosporic embryo sac means only one megaspore take part in the development of embryo sac. Only one megaspore take part in the development of embryo sac. That is called as monosporic type of embryo sac. Is it clear? At the end of meiosis, how many megaspores are formed? 4 megaspores are formed. But in monosporic, out of 4 megaspores, only 1 megaspore take part in the development of embryo sac, that is called as monosporic. If 2 megaspores take part in the development of embryo sac. If 2 megaspores take part in the development of embryo sac, that is called as bisporic. Okay. Moving to the third type, that is the tetrasporic. Tetra means 4, bi means 2, mono means single. Tetrasporic means here all 4 megaspores take part in the development of embryo sac. That is called as tetrasporic embryo sac. Here, all four megaspores take part in the development of embryo sac. Is it clear? So all four megaspores take part in the development of embryo sac, that is called as the tetrasporic type of embryo sac. Coming back to as per the NCERT, they are explained the monosporic type of embryo sac. Why they are given monosporic? Because in most of the flowering plants, monosporic type of embryo sac. Monosporic embryo sac is also called as polygonum type of embryo sac, because this type of embryo sac development first observed in polygonum by the scientist Strasburger. So we go to the female gametophyte. In most of the flowering plants or in angiospermic plants, monosporic type of embryo sac. Monosporic type of embryo sac it is also called as polygonum. It is also called polygonum type of embryo sac, because this type of embryo sac development observed in polygonum by the scientist Strasburger. So it is also called as polygonum type of embryo sac. Also monosporic or polygonum type of embryo sac. Already I told that what is meant by the monosporic type of embryo sac. Single megaspore take part in the development of embryo sac is called as monosporic. So here, the megaspore mother cell, when it undergoes, it gives rise to how many megaspores? How many megaspores are formed? 4 megaspores. How many megaspores? 4 megaspores are formed here. So these are the tetrad of megaspores. This is the what? Tetrad. Tetrad of megaspores. Okay. So in monosporic, out of 4 megaspores, here you can see here, how many megaspores are there? 1, 2, 3, 4. So in monosporic, out of 4 megaspore, only 1 megaspore is functional. Only 1 megaspore is functional. Remaining 3 megaspores are degenerate. These are the degenerating megaspores. Degenerating megaspores, and this is a functional functional megaspore. This is the functional megaspore. So in monosporic type of embryo sac, only one is functional. That is fourth from the micropyle. This is the micropylar region which megaspore is functional, fourth from the micropyle. Fourth from the micropyle. Fourth from the micropyle. Is it clear? It is a very important point. So which megaspore is functional? Fourth from the micropyle. So that megaspore is functional. Now, this functional megaspore nucleus. This is a functional megaspore, and this is the functional megaspore nucleus. It divides mitotically. Careful listen here. It divides mitotically to form two nuclei. Two nuclei. Two nuclei. One move towards micropylar region. One move towards the chalajal region. One move towards the micropylar region and another move towards the chalajal region. So this functional megaspore nuclei divide mitotically to form how many nuclei? Two nuclei form. One move towards micropylar region, another move towards the chalajal region, and this is called as two nucleate embryo sac. Two nucleate embryo sac. Two nucleate embryo sac. Here, nuclear division is free. Nuclear division is free. Because as soon as new formation of nuclei, there is no any cell wall formation. So only it is called as the free nuclear division. Okay, so this is a two nucleate embryo sac. One more mitotic. So first mitotic division, two nuclei formed. In second mitotic division results in the formation of how many nuclei? Four nuclei. Two present at the micropylar region, two are present at the chalajal region. Two are present at the micropylar region and two are present at the chalajal region. So in third mitotic division results in the formation of how many nuclei formed here? In first mitotic division, two nuclei formed. Second mitotic, four nuclei formed. In the third mitotic, how many nuclei formed? 8 nuclei are formed. 4 present near micropylar region and 4 are present at the chalajal region. Is it clear? 4 are present at the micropylar region and 4 are present at the chalajal region. So this is called as the 4 nucleate embryo sac. 4 nucleate embryo sac. And this is called as the 8 nucleate embryo sac. 8 nucleate embryo sac. Is it clear? So this is called as the 8, this is the 4 nucleate embryo sac and this is called as the 8 nucleate embryo sac. Here a very important here. So here nuclear division is free nuclear division. After 8 nucleate stage, cell wall formation occur. Is it clear? After the 8 nucleate stage, cell wall formation takes place. First, 6 of 8 cell is surrounded by 6 of 8 cells are surrounded by cell wall, and organized into cells. So here you can be observe that after the 8 nucleate stage, cell wall formation occurs. 6 of 8 nuclei are surrounded by cell wall and organized into cells. 3 cells are present near the micropylar region, and 3 cells are present near the chalazal region. Okay, and 2 polar nuclei, one from micropylar region and another from the chalazal region. So these polar nuclei come at the center, and these are surrounded by the cell wall, and this is called as the central cell, and these are the polar nuclei. So 3 cells are present at the micropylar region and 3 cells are present near chalazal region, and 2 polar nuclei. Why it is called polar nuclei? Because nuclei come from the polar region, one from micropylar region, another from the chalazal region. When both the nuclei move at the center, so these 2 nuclei surrounded by cell wall, and that is called as the central cell, and these 2 nuclei are called as the polar nuclei. Is it clear? Concentrate here. A very important one is here. Megaspore mother cell undergoes how many mitotic division to produce embryo sac? Megaspore mother cell undergoes 3 mitotic division. Understand here. Megaspore mother cell undergoes megaspore functional megaspore undergo how many mitotic division? 3 mitotic division to produce embryo sac. Okay, sorry. Here megaspore undergoes how many mitotic division? 3 mitotic division. Is it clear? Megaspore mother cell undergo meiotic division to form haploid of or tetrad of megaspores. Is it clear? And functional megaspore undergoes how many mitotic division? 3 mitotic division. Remember it here. Megaspore mother cell undergoes which division? Meiotic division to produce tetrad of megaspore, and in monosporic single megaspore is functional, and that single megaspore nucleus undergoes 3 mitotic division to produce the embryo sac. Is it clear? Okay. After this, structure of mature embryo sac. moving to the mature embryo sac. So this is the structure of mature embryo sac. Okay, cell wall formation takes place after the 8 nucleate stage. Is it clear? So this is the structure of the mature embryo sac. Mature embryo sac is 7 celled and 8 nucleate. 7 celled and 8 nucleate. See here, 3 cells are present near chalazar region and these cells are called as antipodal cells. These are called as antipodal cells. They are present near where? Always chalazar region. So, 3 cells are present near chalazar region called as antipodal cells and 3 cells are present near which region? Micropylar region. This is called as egg apparatus. It is called as egg apparatus. And egg apparatus, it consists how many cells? 3 cells. 1 egg cell, 1 egg cell and 2 synergids. 2 synergies. Is it clear? And at the central cell and these are the 2 polar nuclei. Remember it here. Embryo sac, it is a 7 cell. See here. Carefully see here. How many cells are there? 1, 2, 3, 4, 5, 6, 7. How many nuclei are there? 1, 2, 3, 4, 5, 6, 7, 8. So, embryo sac, mature embryo sac is 7 celled and 8 nuclei. And here you have to remember that egg apparatus always present near the micropylar region. Egg apparatus, it consists of 1 central egg cell and 2 synergids. And antipodal cells are present near chalazar region. Is it clear? And central cell present at the center. Is it clear? Here, a very important, what is the ploidy of the antipodal cell? Ploidy of antipodal cell. Ploidy of antipodal cell. haploid. Ploidy of antipodal cell is haploid. Haploid. Okay. What are the ploidy of synergids? Synergids, haploid. What are the ploidy of egg cell? Haploid. Ploidy of egg cell is also haploid. Then what is the ploidy of the central cell? Here you have to remember it here. Just before the fertilization or just prior to the fertilization, polar nuclei fuse. Just before the fertilization, what will be happen there? Two polar nuclei form. When those two polar nuclei form, that is called as secondary nucleus, or it is also called as definitive nucleus also. It is also called as definitive nucleus also, and this is diploid. Is it clear? This is a diploid. Understanding here, when one male gamete, you know very well, double fertilization, one male gamete fuses with the egg cell to form zygote. Zygote is a diploid because it is a product formed by fusion of haploid male gamete with haploid female gamete. Then N plus N, haploid male gamete and haploid female gamete, result in the formation of what? 2N zygote. But male gamete, when fuses with the secondary nucleus, this fusion is called as what? Triple fusion. It results in the formation of what? Primary endosperm nucleus. And this primary endosperm nucleus is a 3N. It is a triploid. Primary endosperm nucleus develops into the endosperm. Zygote develops into the embryo. So very important, you should remember the ploidy of synergid, ploidy of antipodal cells, ploidy of egg cell, and ploidy of the secondary nucleus. Is it clear? So this is the structure, and a very important one is synergid contain some cellular thickening, and this you are observing the finger-like structure, cellular thickening, and these are called as filiform apparatus. This is called as what? Filiform apparatus. Filiform apparatus. So what is the function of the filiform apparatus? Guiding the entry of pollen tube into synergy. Guiding pollen tube into synergy. Very important. Filiform apparatus present in where? Synergy. What is the function of this filiform apparatus? Guiding the pollen tube into synergy. Is it clear? And this is the vacuole and this is the nucleus. Remember this when you are going to observe from micropylar region to the chalazion region. First will come filiform apparatus, followed by nucleus, followed by vacuole. If the arrangement of structure in a synergy from the chalazion to the micropylar region means, you should go to the vacuole, followed by nucleus, followed by filiform apparatus. Remember the sequence. Is it clear? So this is the structure of mature embryo sac. Is it clear? Moving to the next one is, after this we will go to the, and you can you tell me which is the nutritive tissue in the ovule? Nucellus. Nucellus is a nutritive tissue. After this, we'll go to the pollination. Pollination. So, till now you have studied the formation of male and female gamete. Is it clear? One embryo sac containing how many eggs? Only one egg. One ovule contain one embryo sac commonly, and one embryo sac containing one egg cell. So till now you are studying the formation of male and female gamete. Commonly here in this member, both male and female gametes are male and female gamete are non-motile. Non-motile. So here both male and female gametes are non-motile. Is it clear? These two bring for fertilization. So how the male gamete is transferred to the female? That is through pollination. This through the what? Pollination. So this occur in gymnosperm and angiosperm. Gymnosperm and angiosperm. Because in this member, both male and female gametes are non-motile, so pollination occur. During this time, the transfer of the pollen grains from anther of a flower to the stigma of the same flower or the different flower. So come back to the pollination. So what is meant by the pollination? Pollination means transfer of pollen grains. Transfer of pollen grains from anther to the stigma of pistil. Stigma of pistil is called pollination. The transfer of pollen grains from anther to the stigma of the pistil is called as pollination. Is it clear? So, or you can define like this also: transfer of pollen grains from anther of a flower to the stigma of the same flower or different flower is called pollination. Is it clear? So, endospermic plants have fascinating array, means adaptation. Actually, endospermic plants are evolved. They have some adaptations. So, that adaptations have agencies for transfer of pollen grains from anther of a flower to the stigma. Is it clear? So, here pollination are of two types. One is pollination classified into how many types? Pollination classified into two types. One is actually, as per the NCERT, they are mentioned three types. Here I will write what are those. One is autogamy. Second one is the botanogamy, and the third one is the xenogamy. Actually, on the basis of source of pollen, pollination are of how many types? Three types: autogamy, botanogamy, as well as the xenogamy. Is it clear? Here, before explaining as per the NCERT, pollination form here, we can tell that autogamy and the allogamy. Autogamy is nothing but the self-pollination. Allogamy is the cross-pollination. So, as per the NCERT, You are going to read this. Okay, as per the reference book, pollination, two types, autogamy and second one, allogamy. Autogamy, it is also called self-pollination. Self-pollination means what? Transfer of pollen grains from anther of a flower to the stigma of the same flower. It means that within a flower, pollination takes place. Self-pollination means transfer of pollen grains from anther of a flower to the stigma of the same flower. Stigma of the same flower is called as self-pollination, or it is also called as the autogamy. It means that within a flower pollination takes place, and that is called as the self-pollination. Okay, here what are the characters of self-pollination or autogamy? What are the characters of self-pollination or autogamy? First one is here, flowers are bisexual. Flowers are bisexual. Is it clear? If both male and female reproductive organs are present in a same plant, then only there is a self-pollination takes place. So flowers are bisexual and there is a synchrony. Synchrony of release pollen grain and stigma receptivity. So here there is a synchrony of release of pollen grain and stigma. It means that both of them, they are going to mature at the same time. Then only there is a chance of self-pollination. Synchrony of release pollen grain and stigma receptivity. The maturation of stamens and carpels at same time. The maturation of stamens and carpels at the same time is called homogamy. Homogamy means here maturation of stamens and carpels. Stamens and carpels at same time is called as the homogamy. Is it clear? Even in autogamy, some flowers are open, always open and exposed to environment. Such flowers are called as chasmogamy. Flowers are called chasmogamy. What are the chasmogamy? Open flowers are called chasmogamy. Open flowers are called as chasmogamy. Second one is here clistogamy. Clistogamy. Flowers are bisexual. Flowers are underground. Flowers are closed. Closed flowers are called as clistogamous flower. Flowers are closed, flowers are bisexual, flowers are underground. So it is called as clistogamy. In some plants such as viola, as well as oxalis, as well as commelina. Viola is also called as the common pansy also. It is also called as common pansy. So in these plants, how many types of flowers are there? Two types of flowers. two types, open and closed flower. In the member of the viola, oxalis and pomelina, plant produces two kinds of flowers, open flowers and closed flowers. Clistogamous flower. Clistogamy means flowers are bisexual, flowers are underground and flowers are the closed. Clistogamous flowers are invariably autogamous. They are clistogamous flowers are invariably autogamous. There is no any chance of cross pollination. They do not depend upon the pollinating agent. They are independent on pollinating agent and they have the assumed seed set, means they are going to set the seed without pollinating agent. Remember a very important part. Clistogamous flowers are invariably autogamous because there is no any chance of cross pollination. They have assumed seed set. Is it clear? So this is about the autogamy. Coming to the allogamy or cross pollination. Allogamy or cross pollination. Going to the allogamy or cross pollination. Allogamy or cross pollination. Here, transfer of pollen grains from anther of a flower to the stigma of another flower of same plant or different is called allogamy or cross pollination. Transfer of pollen grains from anther of a flower to the stigma of another flower of same plant or different plant is called cross pollination. Is it clear? Transfer of pollen grains from anther of a flower to the stigma of transfer of pollen grains from anther of a flower of another flower. Transfer of pollen grains from anther of a flower to the stigma of another flower of same plant or different plant is called as the allogamy or the cross pollination. So here, come back to the NCERT. Second type that I told you, the gitanogamy. Are we autogamy or studied? So gitanogamy, and the third, that is the xenogamy. Actually, these two comes under the cross pollination. Is it clear? So what is meant by the gitanogamy? What is gitanogamy? Gitanogamy, transfer of pollen grains from anther of a flower to the stigma of another flower of same plant. Here, transfer of the pollen grains from anther of a flower to the stigma of the same flower is called as the gitanogamy. It brings genetically similar pollen grain. It brings genetically similar pollen grain. It brings genetically similar pollen grain because pollen grain come from the same plant. So only it is a genetically similar. Moving to the next one is here the xenogamy. So what is meant by the xenogamy? Xenogamy means So xenogamy means here transfer of pollen grains. So xenogamy means here transfer of pollen grains from anther of a flower to the stigma of another flower of another plant. Understand here? Xenogamy means here transfer of pollen grains from an anther of a flower to the stigma of another flower of another plant is called xenogamy. It brings genetically different pollen because pollen grains are coming from different plant. Genetically it brings different pollen grain because they are genetically different because pollen grains come from the two different plants. So these two actually included under the way cross pollination. Cross pollination. Cross pollination. So as per the NCERT, how many types of on the basis of source of pollination? There are three different types. One is autogamy, second one gitanogamy, third one is the xenogamy. Autogamy means transfer of pollen grains from an anther of a flower to the stigma of the same flower is called as self-pollination or autogamy. So in autogamy, flowers are bisexual and there is synchrony of release of pollen grain and stigma receptivity. So then even you are studying about the chasmogamy and cleistogamy. Chasmogamy means flowers are open. Open flowers are called as the chasmogamy. And cleistogamy, flowers are closed, flowers are bisexual, flowers are underground. Cleistogamous flowers are invariably autogamous. There is no any chance of cross pollination. They do not depend upon the pollinating agent. Is it clear? In the member of the viola, ovules and camelina, there are two types of plant produce, two types of flowers, open flowers as well as the closed flowers. So closed flowers which are underground and open flowers are the aerial. Coming to the gitanogamy, here transfer of pollen grains from anther of a flower to the stigma of another flower of same plant is called as the gitanogamy. And moving to the xenogamy, the transfer of pollen grains from an anther of a flower to the stigma of the another flower of another plant is called as the xenogamy. So this is the brief introduction about the pollination. Introduction of pollination. So as per the conspectus, so till you have to read the Megasporogenesis to xenogamy for exam. And second one is here, growth of pollen tube towards embryo sac is due to the chemotropic. That you have to remember it here. The growth of the pollen tube towards the embryo sac is chemotropic. Is it clear? Okay. So this completes the pollination. Okay. Write the exam properly and read complete NCERT textbook because in exam all the questions from the NCERT only. Okay. So tomorrow I will discuss paper. Thank you.